It's a pleasure to be here. Thanks for the kind invitation, and it's an honor to be part of the STREAM Consortium. It's really all the meetings are so professionally organized, and I thank Eva and the rest of the consortium for that. I will switch topic completely and talk about NK cells. Anybody here who works with NK cells? No. Yeah. Okay. So I sort of imagined that would be the case, and I decided to make an historical overview and bring you 45 years back in time to the Karolinska Institute where the NK cell was discovered. And when we make discoveries in the lab and students come and knock on the door and want to discuss a new piece of data, and I think this is common to all of you, they rarely come in and say, Eureka! but what they possibly might say is that, that's funny, this is funny, what's going on? And this is exactly what Rolf Kissling at the Karolinska, after spending some frustrating years trying to develop a cancer vaccine, eventually got to say. So his PhD program was about generating a tumor vaccine. The problem is that the control mice were equally efficient in clearing the tumor cells as the mice that were vaccinated with the lysed tumor cells. And it turned out that the background killing was mediated by a new cell type that they eventually termed the natural killer cell. And this is actually a picture of Rolf standing with the PhD thesis, like a roll of paper, outside the tumor biology building at KI. And this is from the manuscript that was published in European Journal of Immunology 41 years ago. And what they are actually editing here manually, as you can see, and coming up to the following conclusion, that the killer cells can be separated from other groups of cells known as macrophages, monocytes, granulocytes, or null cells. It would thus seem fair to state that the present spontaneous anti-myeloid leukemia killer cells thus represent a new, previously undefined cytotoxic cell. So that was a funny discovery. His first student was Claes Kärre in the early 80s. And what Claes tried to do in the end of 70s, early 80s, this is Claes in the lab, he tried to solve one of the most puzzling observations at the time, and that was the hybrid resistance. So the hybrid resistance is the ability of the F1 hybrid to reject a parental graft. And that was really peculiar, as they point out in this GEM paper 71, because there's nothing foreign coming. So if you take spleens from the parent and infuse into the F1 hybrid, that obviously has a more complex background because of the paternal input, it rejected the graft without anything foreign being present. So it's about looking at what everybody looks at, but see what nobody else is seeing, the prepared mind. I think this is a quote from Louis Pasteur. And this is what Claes did. He was, at the time in the 80s, outside the Swedish archipelago. We had Russian submarines coming in, invading the coast. And this is from a Swedish news magazine, not very sophisticated news magazine, but Claes was reading it while contemplating on his experimental data. And the instruction here was how to recognize a foreign submarine. So the people living in the coast, they were taught how the Russian submarines looked like. And there are plenty Russian submarines, as you can see. And they were supposed to call in if they found one of these, and nobody ever called. It's too complicated to memorize all of these Russian submarines. So they switched strategy. Instead, they taught the people at the coast how the Swedish submarines look like. There are only 3 of those. So when you see a submarine and it's not one of the Swedish submarines, then you should call. So then it's probably a Russian submarine. And this is actually a true story. So this was the time when Claes, in his in the library formulated the missing self hypothesis. In his thesis 81, the paper was published 86. So he postulated that NK cell recognition of targets is based on the ability to recognize information that is missing on the target, rather than opposite, recognition of something that is positive, which is what T cells do. They recognize the Russian submarines. And this explained the hybrid resistance. So the recipient is now rejecting the incoming graph because it's lacking D. It's missing the self D. So it's sensing the absence of something that is present in self. And in the formulation of the hypothesis, they predicted that this would be mediated by some sort of receptors that sensed something self on the class 1, on the tumor cells. And that's exactly what it was. So basically what NK cells are sensing is the absence of self MHC class 1 molecules, HLA in the human. And they are doing that by inhibitory receptors that basically turn off NK cell activation when the MHC class 1 levels are present. Obviously, they also actually in that hypothesis predicted the existence of activating receptors. And indeed, you need some sort of stimulation through activating receptor that sends stress-associated molecules expressed on cancer cells or infected cells. So if we focus in on this inhibitory HLA interaction, it's depicted here. This is the MHC class 1 molecule with the peptides. This is the footprint of the T cell sensing the class 1 complex plus the peptide. But the NK cells is sort of sneaking in on the side with inhibitory receptors that bind to specific motifs in the class 1 molecules that are determined by polymorphism at position 80. So there are two major motifs that are the C1 and C2 motifs, and a number of alleles that contain this particular motif. So here's an NK cell expressing inhibitory receptor interacting with the class 1 molecule, leading to phosphorylation of SHP1 and inhibitory signaling. There are a number of receptors, inhibitory killer cell immunoglobulin-like receptor that bind to different groups of class 1 molecules. There are also complementary MHC binding receptors that are important, NKD2A that binds to HLA-E. Then there is also a number of activating KIRs that have less well-defined roles. Possibly they have been involved in viral immunity in earlier days, but it's really not very clear. And this NKD2C receptor that binds to HLA-E, which is a non-classical class 1 molecule. And I'll come back to that in a minute. So genetic studies where people have been looking at epistatic interactions between KIR and HLA have suggested a role for these immune genes in many different clinical contexts, in infectious diseases, in particular HIV and hepatitis C virus, also in reproduction with work from Ashley Moffat at Cambridge, which may in fact be the main driver of this polymorphism. And in cancer with the pioneering work from Andrea Velarde here in Italy, in Perugia, but many others had followed along the same lines, and also in autoimmunity. So if you are working on T cells, you should still pay attention to the KIRs, because they are highly expressed on T cells. This is a paper we published a couple of years back where we looked at the KIR expression on T cells using lots of antibodies combined together, and we can see that there is a gradual increase in the KIR population as the cell matures towards an effector memory RA converted phenotype with the CD57. You can see almost 40% of the RA positive, 57 positive T cells express KIR. And sometimes it's up to 80% of those cells in individuals. So the emerging concepts in it's the title of this talk, and if we look at the last decade, the two key developments in the field are the following. So it's the finding that NK cells are functionally calibrated against the self-MHC. And this is a process with many names. For some reason, we can never make up our minds in immunology, but it's termed education, typically in Europe, and licensing in the US. There are also other models that sort of that contribute to the understanding, arming, disarming, that you might have heard of. I will refer to this process as education. The second key development in the last few years is the fact that NK cells are differentiating, that they live longer than we previously thought. It was believed that they would live only 2 weeks, very short lifespan, boring life, no dynamic, no plasticity. But now we have understood that similar to T cells, they actually differentiate and into terminally differentiated subsets, and some even think that they become memory NK cells. Again, we have many names: memory NK cells, adaptive NK cells, adaptive memory-like NK cells, etc. So we are referring to these as adaptive NK cells. So my lab focuses entirely on 50% on understanding the mechanism behind education, and 50% of the lab basically explore mechanisms of differentiation. It's a golden era to work in immunology now, I think. So this is a flow cytometry staining performed by Niklas Björkström, a former student in my lab who's now having his own lab. It's a 23-color flow cytometry panel of one sample. And you can basically see all immune cells that we have represented from a single staining. So from the NK cell perspective, we have the naive CD56 bright, the dim cells, and also the terminally differentiated NK cells. So zooming in on these NK cells and using mass cytometry, Peter Parham and others have shown that we have 100,000 different subsets of NK cells. Each of us as individuals between 8 and 30,000 subsets, 8,000 and 30,000. So this is at the phenotypic level, but it's been known for a long time that the functional diversity is also very important. And this is from the Nobel lecture in 1960 of Peter Midavar, who was contemplating on the concepts of tolerance. So he asked the question whether an animal is partially tolerant, because all of its cells are almost completely debilitated, or because while most of them are completely functional, out of function, a minority retain full possession of their power, which was not possible to study before. Now we have new tools that make this possible. And I think the most advanced tool is actually developed by a colleague of mine at the Karolinska. This is work of Björn Örnfeldt at KI. There are some other similar platforms around, of course. But what he's been doing is to develop microchips to study phenotypic functional diversity. So this is an NK cell in blue killing tumor cells. So when they are killed, they turn red. The first two are killed by necrosis, really bam, quick hits. The two other tumors are killed by apoptosis. You can see the characteristic blebbing. The mechanism behind this is not known, but you can see diversity in the behavior. But to really make sense of this, he put these on chips. These are produced in Ericsson's old Ericsson. It's an electro company in Sweden. In the old factory, they have access to make these plates. So they can actually screen all these wells automatically in a high throughput to try to understand the functional diversity of the NK cell response. They're also looking at T cells. So to illustrate my point, if you have two NK cells to choose from for a clinical trial, you would rather pick this cell than this cell that is sort of just sniffing on the tumors without doing anything to them. Whereas this one is actually killing everything and waiting for more to kill. Yeah. So I talked about the two different new developments, NK cell differentiation and NK cell education. So NK cell differentiation is depicted here. We're starting with precursors in the bone marrow, and then development towards mature NK cell stage 4 that are bright, that eventually turn into dim cells, and there we have a continuous differentiation to the memory or adaptive cells here. And it's associated with changes in lots of differentiation markers, cytokine receptors, activating receptors, inhibitory receptors, of course, but also effector molecules like perforin and granzymes, and the functional behavior changes. So there's another way to look at it, the functional potential on the y-axis and the differentiation of the cell. Starting off as a naive cell, it's very responsive to cytokine input, like all naive cells are. Eventually they become much more focused on receptor input through CD16, the antibody receptor, but also through NKD2C, which is an activating receptor on the cells. This process is transcriptionally regulated. So you can have this, if you do transcriptional profiling, you can see a gradual increase in some transcripts and a decrease in others. And it's also regulated at the epigenetic level, in particular this final stage here. But on top of this, the cells acquire receptors, the KIRs that I spoke about, and they do so in a stochastic manner. So some cells at this very stage of development, when they start to express KIR, you will have some that expresses self-receptor and some that expresses non-self-receptor. And it will put them on different functional trajectories. So this cell is much more responsive than this cell. And this is not regulated by transcription. And nobody really knows how this works. And I'll show you some data how critical this is for the function of the cell. So that's the next part of the talk. So NK cell education, we call it the writing on the cell wall. What receptors are actually expressed at the cell surface. And this is how it works. So you have an educated NK cell that expresses a KIR2DL1, which is specific for HLA-C2. So in this context, there's inhibition. It's not going to be killing this normal self cell. And now let's assume that this cell is transformed to a tumor or infected with a virus, so it downregulates class 1, the missing self, the lacking Swedish submarine. This NK cell will then sense this absence of self, go to the target, form conjugate, and kill it, as illustrated in the ability of the cells to degranulate here. We have another cell that expresses KIR2DL3 in the very same donor. So it's interfering with, it's not recognizing the C2. So when it's circulating in the body, there is no inhibition ever taking place. So if this would be fully functional, it would kill in a missing cell fashion. So that's not okay. So the way this is dealt with is basically to cause hyperresponsiveness. So for the cell, for the NK cell, this is exactly the same as this, because it's not binding to that class 1 molecule. Are you with me? This is the same. So when this happens, it's not change, it's not sensing a change, and therefore there is no response. So this almost digital difference between functionality in the cell is not caused by transcriptional differences or by any other known mechanism, and still is obviously quite striking. So what we want to do in NK cell biology is to try to understand how we can exploit these cells for cancer immunotherapy. And we think they have a niche to fill. And that relates back to the excellent introduction by Jakob earlier on escape variants. So the key challenge I think that we all face and that we try to address by different, either inhibiting RNase or other means, is to convert immune failures to clinical responders. And to do that, we need to know exactly what's going on. And there are very few studies on that have explored that as of yet. This is one that was published earlier this year, where 4 patients that were treated with 4 patients with metastatic melanoma that were treated with checkpoint inhibition anti-PD-1, and they were refractory to the therapy. So they looked at the tumor before and after. And they found in those 4 patients that one of them had a point mutation in beta-2 microglobulin, leading to complete loss in class 1. 2 others had mutation in JAK1, I think, leading to failure to respond to interferon and upregulation of class 1. So at least for 1 out of 4 in this first very comprehensive study on the resistance to checkpoint have lost class 1. So if that number actually holds, so it's 25% of these cases, that would be the niche for NK cells. Because remember, NK cells, they kill when class 1 is lost. So if you have the responders and the non-responders, loss of class 1 may be one mechanism, and then there are a number of other mechanisms, of course. And the purpose of NK cell therapy would be to maybe convert these failures into responders. So the next generation NK cell therapy, based on the insights that we have generated in the past decade. I told you that NK cells differentiate from naive to more differentiated cells. And if you just take NK cells from any donor and stimulate with cytokines, we would grow out these naive cells. These are the ones that will respond to cytokine input. So what we are trying to do in the lab is to generate strategies to get these cells to grow. And that can be done either by some sort of guided differentiation, where we know how this program is regulated so we can push it forward, or by platforms that allow selective expansion of the cells of interest, maybe these educated cells or the adaptive cells. And so to do this, one might get clues from viral immunity, how NK cells are responding to viruses. And a number of years ago, we looked at NK cell repertoires in healthy donors like ourselves. We looked at 200 donors using a 14-color flow panels with 7 different KIRs. These are 48 of those donors. They are all negative for cytomegalovirus. And they have this repertoire. This is 7 KIRs. And you can combine those stochastically, if you remember the mathematics, into 128 different variants. So to the left, we have cells that express all 7 KIRs. And according to probability rules, that's very unlikely to have all 7. And indeed, you don't see any cells, the frequency is 0, having all 7. Over here we have cells that express only 2DL1, and it's quite common to have only one receptor. So this really follows the rules of probability. So we had this distribution. We identified some donors that looked like this. So here is a perfectly healthy donor coming to the blood bank and donating blood. We go to cells, and 75% of the NK cells have only one KIR expressed. And it's a KIR that recognizes HLA-C2, so it's a self-KIR. And we only see this type of expansions in patients or donors that are cytomegalovirus positive. So this is a response to the cytomegalovirus. And here's another donor. It's 2DL3 expanded, it recognizing self. So we thought maybe we can mimic CMV in vitro. And basically, this is what I told you about. We have a donor and the recipient with leukemia, where there's a mismatch in the class 1, exactly what Andrea Velarde was studying in transplantation. You will have some of these cells that have a self-receptor that are educated in the donor. They're functional, they're cells that kill a lot of targets. And if you transfer these to the recipient, they can possibly mediate anti-tumor responses. We have just finished a clinical trial at the Karolinska with 16 patients receiving this type of cells. But now we're talking about the next generation NK cell therapy. So what cytomegalovirus is doing in some of us is to expand large numbers of these cells. And we thought that if we can do this in vitro, we can maybe get a better clinical product for therapy. So drivers for these adaptive responses are sort of known, at least to a certain extent. And one is the NKD2C receptor that binds to HLA-E, as I told you about. We also identified some potential role for activating KIRs in this response. So by basically mimicking CMV, we want to generate a platform where we can get this scenario going, where we can stimulate NKD2C and get cells to proliferate. So selective expansion of adaptive NK cells. And there are a number of platforms available for doing this. One is, as I say, using a feeder cell that expresses HLA-E to stimulate NKD2C. You can have exactly the same constituents, but do it on a bead or in some sort of plastic-coated surface. Alternative, one can stimulate through activating KIRs, and now more recently we identified the role for CD16 and CD2 together to drive this response, which I'm not talking about. So I'm soon ready. I just want to illustrate that this is indeed possible. We've generated data using this platform, and we're working together with potential industrial partners to bring this to the clinic as well. So this is a selective expansion going from day 0 to day 14, accumulating at day 14 a population of cells that are NKD2C positive and negative for the inhibitory NKD2A. And these cells have a unique KIR profile, so they express only 2DL3, I mean at least most of them. This is the accumulated data from many donors from day 0 to 14, how we enrich for NKD2C positive cells that have a self-specific KIR expressed on the surface. And when we take these cells and test them against primary leukemic blasts from patients with acute lymphoblastic leukemia, we see that they are highly potent. So we've never seen this type of killing before, and we're really excited about that. These are primary blasts right out of the bone marrow from patients with ALL. This is a C1C1 patient. So we have two different cultures, one from a C2 donor, the 2DL1 culture, 14 days, and we kill almost all of these blasts. The very same cells, when tested against C2 blasts, are not capable of killing. And the reverse then for the 2DL3 culture that is incapable of killing the C1, but do kill quite efficiently the C2 blast. This is summary of the data, the alloreactive killing and the sort of specificity that they're not capable of killing when the class 1 is there. So just to conclude with some scientific and logistic bottlenecks that I think we're facing in immunotherapy. We still have an incomplete understanding of the development, the homeostasis, homing, and functional regulation of NK cells, so we still need to do more biological science to, for instance, understand how education works. We are struggling a lot, and that's part of the things that we're trying to solve in the STREAM Consortium, how to efficiently deliver genes into primary NK cells. The scaling of GMP-compatible off-the-shelf NK cell expansion protocols is non-existing at this day. We also have problems with getting suitable animal models for testing toxicity and efficacy. Logistic bottlenecks, I guess. Very often this type of advanced cell therapy program are disconnected from the clinical programs, which is a problem because we need to link it up to the clinical care and the good. So that was this point. The sort of the strict response criteria that are used at the national and international level. Obviously, to transfer these cells across institution is also going to be a challenge, and it's very costly. So I'd like to thank the people in the group. So I have a group still at the Karolinska now since a few years out in Oslo. And this work has been done mostly by actually Vivienne and Lisa on the adaptive cells, and the rest is more cartoons. Okay, thank you.